So hello guys and welcome back to Excel Academy. So we were doing translation. So before we proceed with translation, there is this question which I would like to discuss. So Akshay, this is a question, right, which you were facing problem?
Yes, ma'am.
Okay. So, so a molecule of composition, this is given. So we can see that it is poly A strand and poly T strand is given. It's replicated in a solution of adenine nucleoside triphosphate with all its phosphorus atoms in the form of radioactive isotope. Okay. So which of the following statement is correct for daughter molecules to be radioactive? Only the DNA molecule that use the poly T strand as a template would be radioactive. Okay. Only the DNA molecule that use a poly A strand as a template would be radioactive. Okay. Both of the daughter DNA molecules would be radioactive. None would be radioactive. Okay. So obviously 3 and 4 cannot be the answer, right? Now the question is, कहाँ पे confusion आ रहा है? So Akshaya, according to you, what is the answer?
One, ma'am.
Okay.
Using polities.
Yes, I mean, it's actually very obvious. I don't know why in the back it was given poly A, right? See, if we use poly A as template, then on the daughter strand, we are going to get only T, right? And this is not going to be radioactive because it is already given that ATPs are radioactive. So obviously, when you're using poly T strand as template, the daughter molecule is going to be radioactive because that is going to have all your AA, and A is radioactive given in the solution. Okay, so it was actually easy, just got confused because in the back it was given 2, right?
Yes, ma'am.
Okay, good that you guys are actually started doing the questions. So actually there was one more question, right? But the thing is that the graphs are not clear. So I was not able to get the answer, dots and all which they were given. So it was not clear. I'll try to get from the proper CSIR question paper so that the graphs like they have mentioned, right? ऐसे mention किया हुआ है, but I'm not able to identify ये ऐसा कहाँ पे है, ऐसा कहाँ पे है. So which one is A and B? It is not clear to me. So I'll get the proper question. Once the graph is clearly visible, then I'll discuss with you. Okay.
Okay, ma'am.
So what about others? Tanvi, Shruti, are you guys also doing question? I mean, you should do. Otherwise, आपको कभी पता नहीं चलेगा like how much prepared you are.
Yes, ma'am.
Okay. So I am always telling start doing all these questions because ये सिर्फ अभी मैंने मोल बायो का भेजा है. Slowly, the other are also there. There is one entire textbook is there. Which is having the previous year question paper. और सिर्फ previous year question paper करके भी sufficient नहीं होगा, right? We need to do more practice from the different sources as well. Okay, no problem. So let's continue then our lecture today. Okay, so just to summarize like what we have discussed in the previous lecture. So we have discussed about how the tRNAs are getting charged. Okay, so charging of tRNA we have discussed. We have also discussed like how mRNA is present. We do have discussed like how mRNAs are getting scanned also. Can anyone tell me how the mRNAs are getting scanned? Anyone? I know thoda sa gap aa gaya hai, but I'm sure this you guys will be remembering. So in the case of prokaryotes and eukaryotes, there is a difference in scanning of the mRNA. Can anyone tell me what is the difference? Okay, I'll give you the hint. Now anyone can discuss? Akshay, can you tell?
The RPS ribosome binding sequence, the shine diagonal sequence.
Yes.
So they scan that. I'm not sure, ma'am. I don't remember it exactly.
BS is present on the your mRNA. ठीक है, prokaryotic mRNA it is present. If you remember the figure, हम कैसे बनाते थे? 5-UTR, then RBS is present, and then comes your start codon over here. So this RBS is nothing but your shine Dalgarno sequences, correct? Now this is going to form complementary base pairing with the 16S with 16S rRNA. Okay, this is how they start the prokaryotic translation, whereas in the case of eukaryote, the ribosome is going to scan the 5' methyl cap of your mRNA. Itna sa difference hai. Okay, so whenever you study anything in mole bio or anywhere, we need to remember always the difference between the prokaryotes mein kaisa hota hai and eukaryotes mein kaisa hota hai. Okay, okay, so we have discussed this also. mRNA mein ribosome structures also we have discussed, like 30S subunit is composed of what and 50 subunit composed of what. Similarly, in the case of eukaryotes also 40 and 60 you need to know. Now why it is 80, why it is 70 also we have discussed, correct? Now let's proceed further, and yeah, of course with the tRNA also we have discussed. There is something called iso-accepting tRNA. Even this we have discussed, if you guys remember. So iso-accepting tRNAs means what? So the tRNA is going to have different codons, okay, but they are charged with the same amino acid. Okay, so that is your iso-accepting tRNAs, and this is the most crucial function of whom? ये किसका main काम है? The aminoacyl-tRNA synthetases. Aminoacyl-tRNA synthetases. Okay, so we also know that they are of different classes 1 and 2. Difference क्या है? Class 1 means they are binding at the 2-OH, 2 means 3-OH at the acceptor arm of the tRNA. Okay, so that's all for the like summarize we have discussed. Now, ribosomal RNAs are both structural and catalytic determinants of the ribosome. इसका क्या मतलब हुआ? Now, if you see the structure, as I already mentioned that now we have the total available structure of the RNA. Now very easy we can understand like what exactly is happening inside the ribosome. Okay, so if you see, this is your 50S subunit and this one is your 30S subunit. Now this is a front view. Okay, this is a front view and this is the side view. Now can you tell me what peculiarity you see in this figure? Which is the important information, which is going to be important information for us to study further about the translation? Yes, तन्वी देख के क्या लगता है? फ्रंट व्यू में तो इतना कुछ पता नहीं चल रहा है, बट साइड व्यू में हमें कुछ तो पता चल रहा है. So what can you understand from this figure? Anyone? Shruti, would you like to try?
Maybe the active side, the region where the genetic material goes through, which is not seen in the front view.
Huh. So this gap which you see over here, it gives an information to you that this is the area where the translation could happen. Okay. So अभी मैं पता नहीं exactly कहाँ हो रहा है. So ये एक एरिया जहाँ पे हो सकता है. अदरवाइज यू कैन सी देयर इज नो गैप प्रेजेंट. कहाँ पे आपका tRNA आएगा, कहाँ पे आपका mRNA आएगा. So all this confusion will be there. So once they got this view, they understood that this gap is actually sufficient enough for mRNA as well as 3 tRNAs to come and sit. Okay, so this information we get from the side view of your ribosome. Okay, so the core functional domains of the ribosome, that is the peptidyl-transferase center and the decoding center. So these are coming under the core functional domains of the ribosome. So they are mainly composed of either the RNA or mostly the RNA. So we also know that, right? That ribosomes are made up of proteins as well. Now, portions of some ribosomal proteins do reach into the core of the subunits where the function seems to be stabilized the tightly packed RNAs by shielding the negative charges of the sugar phosphate backbone. So this, guys, is very, very important. Like, why do we need ribosomal proteins? Even though we saw that the entire catalytic activity of ribosome is done by the peptidyl-transferase, which is actually in nature the ribozyme. Okay, self-activating or self-cleaving RNAs they are. So हमें proteins क्यों चाहिए? So why these ribosomal proteins are required? So one of the main reason is that we know that we are going to bring the tRNA as well as mRNA, right? And then we have RNAs also of your ribosomes. So तीनों तरह के RNA is coming, and all these three are made up of sugar phosphate backbones, right? Even though they are single stranded, but they do have their sugar phosphate backbone, correct? Now because see, ribosome में आप tRNA भी ला रहे हो, mRNA भी ला रहे हो, so because of the presence of this negatively charged sugar phosphate backbone, what could happen actually? क्या हो सकता है? Just because all these negative charges of sugar phosphate backbones are coming together, what will happen? Yes, Akshaya, क्या होगा?
Repulsion.
Yes, very good. So there will be a repulsion. अब repulsion होगा, then it is very difficult, right? It is difficult. Then in order to carry any activity. So in order to stabilize this negative charge, in other words, we can say that in order to prevent the repulsion between the tRNAs, mRNAs and RNAs backbone, we need these proteins. तो अब यहाँ पे आपको क्या idea मिलना चाहिए? That all the proteins of the ribosomes are then positively charged. ठीक है? Or like you can say that they are rich in basic amino acids. So now in future, if everyone asks you like why the ribosomal proteins are basic in charge or why they are positively charged mainly, okay, then you should know the answer. It is because to prevent the repulsion which is carried between the tRNA, mRNA, RNA, since because of their sugar phosphate backbone, which is negative in charge. Okay, so this slide is clear to everyone. This is important to understand.
Yes,
Okay.
ma'am.
Okay. Now, so ribosome has 3 binding sites for tRNA. ये बार बार repeat होगा because this is something which we need to understand. Okay. एक चीज और मैं बताती हूँ, like if you see in the figure, you won't see that ऐसे gap बने हुए हैं ऐसे. Okay, there is no such gap which is present on the 50S ribosome. It's just that they have got that much space to allow for your EPA site because all these three can be together. Okay, एक साथ वो present होते हैं on the mRNA. Now one more thing which is important over here to understand is, if you see the mRNA ka structure on your 30S subunit every time, okay, this is the cartoon representation, but if you see the 3D structure, like the crystallography se jo structure aata hai, you will also observe one thing, and that is that your mRNA, whenever it is present, can you see a kink which is present over here? When I say kink, iska matlab kya? Koi bhi structure agar aisa hai, you might see something like this. Now, in this structure also, can you see that kink which is present on the mRNA? Yes,
Yes,
right.
ma'am.
Now, can you tell what will be the significance of having this kink in the structure? क्यों है? Moreover, if you see very closely, this kink is specifically present for P and A, but not for E. Okay. तो क्या लग रहा है? क्या reason हो सकता है?
For addition of amino acids.
For what amino acids?
Addition of amino acids, like to add amino acids, it would be helpful.
Add होंगे. पॉलीपेप्टाइड चेन आपका यहाँ से निकलता है. ओके, देन गेस करो. और यू आर ऑन द राइट ट्रैक दो. एमिनो एसिड्स ऐड करने के लिए व्हाट इज द फर्स्ट थिंग व्हिच शुड बी डन ऑन द mRNA राइबोसोम?
Distort the structure.
To
Distort the structure in such a way to enable the adding of amino acids.
नहीं, अगेन mRNA, tRNA, RNA. mRNA, tRNA क्या करते हैं? They do the decoding of the codons, correct? So when this kink is present, it means they are making sure that it is read in triplets only. तो जैसे ही आपका ORF आइडेंटिफाई हो जाता है, you get your start codon, ठीक है? Your first amino acid will be there. Now they make sure that after that एक दो तीन के बाद there is a kink and then another 3 will be present over here. So when this moves, again 1 2 3 and then new 3 codons will be read. समझा अभी? Since we don't have any comma or separation between the nucleotides so that it becomes easy for them to understand, so what the nature has adopted is that जब भी it is making interactions, ठीक है? So first your tRNA comes at A site. Let us say that your tRNA is coming, which is having one amino acid, okay? So it is making 1, 2, and 3 base pairing, codon, anticodon, correct? mRNA ka codon, anticodon of your tRNA. So they have made 3 base pairing. Now what happens is that it is going to shift to this side. So when this shifts over here, so what will happen? ये यहाँ पे आ जाएगा, आपका tRNA यहाँ आ जाएगा, correct? Here your new tRNA will come, making sure that इस 3 nucleotide के बाद there is a kink and it is available for base pairing with the another tRNA. Is this much is clear? Like why there is a kink present in the mRNA? Okay. And if the question comes like E के पास क्यों नहीं है? So E means what? It is the exit of the tRNA. So obviously there we don't need any complementary base pairing with the mRNA because it has already read or decoded the codons, transferred the amino acid. Now it is ready. Now it is pre-tRNA. So it does not need any interactions with the mRNA, and that's why it is not in connection. Okay. Okay. Now look at the structure. This is the structure where you can see that the entire ribosome, okay, along with your tRNA and all the three tRNA they have shown. Like it is bound with all the 3 tRNAs. Now first one is your front view. Okay, you can see that you have got 3 tRNAs. Though you can't see now the interactions between them, but at least this much is clear that at one point all the 3 tRNAs are bound with the ribosome. Okay, so this one 1, 2, and 3. So this is your front view. Okay, now coming to the second one, this is your side view. So again you can see that the gap which was seen earlier, now over there your 3 tRNAs are present. Clear? Now look at these structures. What do you understand these structures? What is C and what is D? If you have understood A and B, then you can very easily say about C and D. So yes, what is C and D? Yes, Tanvi, would you like to try?
Ma'am, in C, when 2 sides are green and red are closer to that circle.
Okay, but that is not what I'm asking. What is this happening over here? This circle means where the mRNA interaction is happening. Okay, so just apart, just don't think about this circle. What is this entire structure telling about you? Akshaya, you?
Is it a cross section, like to understand like how the tRNAs interact?
Yes. See, what you can see over here is that this is only your 50S. Okay. And then you can see in the 50S how these tRNAs are present. Circle ka matlab hai jahan pe they are making interactions, okay, with the mRNA and all. Now over here, if you see, this is what your 30S. Now you see how much portion of the 30S is interacting with the your tRNAs. It's negligible. अगर हम इसको थोड़ा zoom out करेंगे, then you can see actually. Sorry, zoom in करेंगे, then you can see how whether like what kind of interactions it is making. So again, I would like to suggest you download PyMol. Okay, download PyMol and get these structures from the PDB, protein data bank. So if you just put the structure of your ribosome bound with tRNA, ऐसा कुछ डालोगे, E. coli डाल दो, no problem. Then take that, go to that site, okay, open, वहां पे आपको download का एक option आएगा, उसमें एक फाइल रहेगा जहाँ पे .pdb ऐसे कुछ तो लिखा रहेगा. Download that file, upload into the PyMol. Now, once you upload into the PyMol, you will get the entire this structure. What you can do is from the right hand side, just click ribbon and ball structures, then zoom in. तो आप देख सकते हो वहाँ पे वो कैसे interact कर रहा है, like how much gap is present. So they will show you if this is the anticodon of the tRNA and this is your mRNA, the distance between these two, they will also tell about it. Okay, so just for your practice, it is good to know about the PyMol. So it's a free software. That's why I'm telling you. You can just download this and visualize the structures. It's not only about the ribosome. Any protein structure, if you want to visualize, you can do. Just go to the PDB, download the file and upload in the PyMol. अगर आपके पास टच स्क्रीन वाला लैपटॉप है, more easy. You can just, with the fingers, you can just rotate the structure and see the interactions, or you can just zoom in, zoom out, टेढ़ा मेढ़ा जो करना चाहते हो आप कर सकते हो. Okay, just try this one and let me know, are you able to do or not, or maybe I can take a session on this, like how exactly to do. Okay, so just let me know, but try to do first by yourself. Okay, so as already said, there are 3 binding sites. So A site is for the aminoacylated tRNA, यानी कि जहाँ पे नया आपका charged tRNA is coming. P is for your peptidyl-transferase, peptidyl-tRNA. E side is binding site of the tRNA that is released after growing the polypeptide chain has been transferred to the aminoacyl-tRNA. So E stands for exiting. Now, each tRNA binding site is formed at the interface between the large and the small subunits of the ribosome. Yes. So in this way, the bound tRNA can span the distance between the peptidyl, transferase and the decoding center in the small subunit. ठीक है, ये clear है ये sentence? So the bound tRNA can span the distance between the peptidyl transferase center and the decoding center. Now the 3' end of the tRNAs that are coupled to the amino acid or to the growing peptide chain are adjacent to the large subunit. इसका क्या मतलब? So they are saying that you have got your 30S subunit, you have got your 50S subunit. Okay, so they have scanned in such a way that, let us say that we have got the mRNA also. We have got the mRNA also. Now these tRNAs are coming. So पहले आपका वो A site में आएगा, correct? यहाँ आपका ये P site हो गया, ये E site हो गया है. Now it is making sure that at A site that it is making a proper codon anticodon base pairing. Okay, and this is present next to your peptidyl tRNA subunit area. Okay, I hope this much is clear. Now there are certain channels which is present on the ribosome which will allow your mRNA as well as the growing polypeptide chain to enter or exit the ribosome. So yes, this is also required to be understood by us कि आपके 50S and 30S में a space should be there so that your mRNA can exit, and on the 50S subunit you need to have a channel from which the polypeptide chain can also exit. Okay, so again, so once you have this 3D structure of the protein, then it becomes easy to understand कि कहाँ पे वो channel या tunnel present है जहाँ से आपका mRNA हो गया हो या polypeptide chain हो गया हो, it can be removed. Okay, now before I proceed, if as far as I remember, there was this question last time I have asked कि ribosome mRNA structure में, is it the mRNA which is moving or is it the ribosome which is moving? What do you think, guys? क्या है इसका आंसर?
Ribosome.
Okay. What about others? Yes. तन्वी, श्रुति, तुम्हें क्या लगता है क्या होगा आंसर? ओके, क्यों लगता है राइबोसोम इट इज मूविंग? As far as what we have discussed till now, क्यों ऐसा लगा that ribosome is moving? The same question is for the mRNA also. If you think mRNA is moving, ऐसा क्यों लगता है? Yes, I want all 3 of you to answer. तो कोई भी पहले शुरू हो जाओ. I'll go alphabetical order. Akshaya, yes. What is your justification?
I feel the ribosome is decoding, like so the ribosome will I think so.
Okay. Shruti?
I think that it's more can probably move a lot better than mRNA. So
What do you think? Just say it again.
ribosome is more compact compared to the long RNA.
Huh?
So
So,
it might be more efficient for it to move compared to mRNA.
okay. Tanvi?
Yes, ma'am. Maybe ribosome is moving uh, in the growing polypeptide chain. No, I mean, ribosome is moving, and because
Because?
ma'am, like
Okay, but तुम तीनों ने राइबोसोम मूविंग बोला है, ठीक है? अब एक चीज, एक छोटा सा इंफॉर्मेशन मैं और देती हूँ. So, have you guys know about that there are two types of ER, endoplasmic reticulum?
Yes.
मालूम है? Okay.
Yeah.
That is, what are those two types of ER?
Smooth and rough.
Smooth and rough.
Very good.
then
Endoplasmic reticulum, and then we have got rough endoplasmic reticulum. Okay. Why it is called smooth and why it is called rough?
Yeah,
rough
presence of
because
ribosome.
presence of ribosome.
One at a time. Okay. Rough it is called because?
Ribosomes are.
Correct. क्योंकि ER में आपके राइबोसोम्स बाउंड है. करेक्ट. अब भी तुम्हें लगता है that it is the ribosome which is moving, because in the case of the RER your ribosomes are bound over there, they can't move. अब किसको मूव होना पड़ेगा? बट यहाँ पे भी आपका पॉलीपेप्टाइड चेन आ रहा है. ओके, यहाँ पे आपका mRNA आता है, राइबोसोम तो फिक्स है, तो कौन मूव हो रहा है? So now it is your RNA. ठीक है? Okay, so before we proceed in the translation, मैं आपको एक थोड़ा सा आइडिया दे देती हूँ, like how exactly this is happening. You need to have a very what to say, broad, you need to keep a broad mind so that you guys can imagine, because it किसी भी textbook में नहीं दिया है. It's just that with, you know, when you read 4 to 5 textbooks were written by different authors, okay, of course the foreign authors. So like हमारे कॉलेज में होता था कि हम हमेशा फॉरेन ऑथर्स ही फॉलो करते थे. ऐसा नहीं है that I'm against Indian authors. It's just that the clarity of a concept is given very beautifully by those foreign authors textbook. Okay, so अभी आप ऐसे देखो, what exactly happens is that if you talk about rough ER, तो rough क्यों है? Because of the ribosome. Correct. Now अब कैसे होता है? If you remember in the previous lecture, हमने क्या बोला था? First it is your mRNA which comes in bind to the 30S subunit and then it is your 50S subunit. अब बाउंड राइबोसोम में जो आपका एंडोप्लाज्मिक रेटिकुलम में जो बाउंड है उसमें कैसे होगा? So always what is happening is that first it is your 30S subunit which goes in bind to your mRNA, फिर आपका tRNA आता है, उसके बाद फर्स्ट tRNA आता है, उसके बाद 50S subunit आता है. Now this entire complex goes and binds to your endoplasmic reticulum if it is bound ribosome की बात कर रहे हैं, और जो फ्री है वो तो साइटोप्लाज्म में प्रेजेंट है. ओके, नाउ दिस मच इज क्लियर कि कैसे. तो ऐसा नहीं है कि ER में अगर 100 राइबोसोम है तो वो 100 के 100 ही प्रेजेंट होते हैं. नो, वंस द ट्रांसलेशन इज डन दे गो बैक. It's just that those proteins needed to be present directly into the lumen conditions. The pH जो होता है आपका ल्यूमन का, lumen का, it should be present directly into that. That's why it is ऐसा कुछ है कि bound ribosome का जो protein रहता है वो directly lumen में जाता है. Okay, otherwise it can be released into the cytoplasm, no problem. Now it has been done. Okay, आपका translation हो रहा है. Now in the translation during the elongation, it is the tRNA. If you guys remember, always we have seen the structure of the tRNA कैसा कि एक ऐसा है with the bound amino acid and एक ऐसा है आपका with the bound amino acid. Now this is there. Now why this structure is important? Because so that this amino acid can be transferred over here, correct? So that this peptidyl-tRNA can become E and this amino acid-related tRNA can become ptRNA, if you remember. Now when the structure is like this, ठीक है, जब ये आपका tRNA ऐसे है और ऐसे जब होता है, that time what it does is that it pushes your codon, okay, ऐसे लात मारता है वो उसको. मैंने ऐसे kink दिखाया था, correct? So when these interactions are there, when there is a twist in the tRNA, it gives a ऐसे jerk, एक तरह से ऐसे लात मारता है आगे की तरफ. So what happens? This tRNA, this portion of your codons gets shifted to this side, and then another 3 codons comes. थोड़ा सा idea आया कैसे? If I try to, I'll try to get the images or videos for you guys, but are you able to get it? It is a structural change of your aminoacyl-tRNA. When it is becoming to peptidyl-tRNA, there is a structural change, okay? And that structural change is making your mRNA to get kicked out. ठीक है, तो वो आपका 3 कोडॉन से और शिफ्ट हो जाएगा. So it is always your mRNA which is getting moved. आप ये देखो ना, मैंने किसी चीज को होल्ड करके रखा है. Let us say that this tRNA is holding the mRNA with the help of the complementary base pairing between the codons and anticodons. Now when there is push, okay, मैं पीछे से या आगे से जो भी हम कहते हैं ऐसे jerk मारूंगी तो what will happen? Those 3 codons will move ahead so that I can move to the next base. And आपका A center जो है वो खाली हो जाएगा. P वाला जो tRNA वो E को पुश हो जाएगा, ठीक है. And then the tRNA which is free of amino acid will be removed. A वाला मेरा P में आ जाएगा. मेरे पास अभी A center जो है वो खाली है. Now the new tRNA will come with the charged amino acid. Okay, are you guys got the point? थोड़ा सा imagination कुछ तो clear हुआ होगा, like how the translation is happening. थोड़ा सा idea आया? थोड़ा सा भी नहीं आया?
Yes, ma'am.
Little imagination is required. Anyways, abhi kaise hai, you have not seen the figures, that's why maybe not able to imagine that much. But I'll tell you, so there are certain elongation factors which helps in the structural changes in the tRNA from A to P that pushes your mRNA towards the outside, and that is how your mRNA is moving and not the ribosome. Okay, chalo. Okay then. Okay, so coming back to the topic, we were discussing like on to the ribosome, we have got the channel and through which by the polypeptide chain can exit and mRNA can enter. Okay, so both the decoding center and the peptidyl transfer center are buried within the intact ribosome, जो center पे रहेगा. Okay, जैसा कि हम दिखाते हैं 30-50 EPA, ये जो पोर्शन है आपका जिस सेंटर में रहेगा. Yet mRNA must be threaded through the decoding center during the translation and the nascent polypeptide chain. Nascent मतलब एकदम newly synthesized. It must escape the peptidyl transferase center. Now, how this has been achieved? So the answer is provided by the structure of the ribosome, which reveals that there are tunnels which are present in the ribosome, in and out of the ribosome. So mRNA enters and exits the decoding center through 2 narrow channels in the small subunit. So obviously small subunit यहाँ से enter कर रहा है तो यहाँ से बाहर भी निकलना है उसको. The entry channel is only wide enough for unpaired RNA to pass through. So what does it mean? तो entry वो जो हम tunnel की बात कर रहे हैं, it is having space only to allow your unpaired RNA. See क्या होता है maximum cases में? Because of the complementary base pairing, okay, there are chances that the RNA may they may form hair loop structures like this. Suppose ये 5' mRNA है, ये 3' आपका poly A tail है यहाँ पे, okay. Now when it is entering, what will happen? There are chances that आपका यहाँ पे base pairing हो जाए, ठीक है. These are the secondary structures which are formed in the RNA. but this is not required. तो ये avoid करने के लिए what they make sure is that the entry channel is only wide enough for unpaired RNA. So सिर्फ single stranded RNA ही enter कर सकता है. अगर ऐसा कुछ बना है, so this will be broken. Those temporary hydrogen bonds will be broken, and then only this RNA can enter the 30S subunit. Okay. Now, this feature ensures that mRNA is in single-stranded form as it enters the decoding center by removing any intramolecular base pairing interactions that may have formed in the mRNA. Okay, so this is what it is doing. So this channel for your entering and exiting of the mRNA is making sure that the mRNA is present in only single-stranded form and there are no intermolecular base pairing interactions present in the mRNA. Okay. Now, so in between the two channels is a region that is accessible to tRNAs, where adjacent codons can bind to the aminoacyl-tRNA and peptidyl-tRNA in the AP sites respectively. Obviously, right? जब आपका 30S subunit में RNA है, तो इतना तो exposure चाहिए to the tRNAs which is present on to the 50S subunit, so that the decoding of the codons can be done. Interestingly, there is a pronounced kink in the mRNA between the two codons that facilitates maintenance of the correct reading frame. So, जैसे कि हमने पहले discuss किया था, so that kink is important. Okay, so जो already base paired है with the peptidyl and the new codons which is going to get paired with the aminoacyl-tRNA codons, anticodons. Okay, so guys, now is it clear why the mRNA is having this kink present?
Yes.
Okay. So this kink replaces any vacant A site codon in a distinctive position that ensures that incoming aminoacyl-tRNA does not have access to the bases immediately adjacent to the codon. So it is making sure, ठीक है, कि इसके साथ कहीं गलत कोडिंग ना गलत पेयरिंग ना हो. So it will make sure कि इसी के साथ ही करे. ठीक है, उनका साइट जो है, the space which is provided to the aminoacyl-tRNA is such a way that it is just going to base pair with these codons only, ना कि उसके बाजू वाले के साथ, और ना कि breaking the hydrogen bonds over here or disturbing the p site tRNA. Okay, so the structures is made in such a way that it is not going to disturb the arrangement of the codons. Now, a second channel through the large subunit provides an exit path for the newly synthesized polypeptide chain. So obviously, right? So धीरे धीरे जैसे ही the amino acids are getting transferred from P to A, okay, slowly it is going to grow in the size. So obviously that needs to be removed from your ribosomal structure. So as with the mRNA channel, the size of the peptide channel limits the conformation of the growing polypeptide chain. So what does it mean, this sentence? It means that it has got more space compared to the mRNA channel, because in order to stabilize the polypeptide chain, many proteins which carry the information of forming the secondary structures, they can form helix. Okay, alpha helix उसमें बन जाता है if it is provided in the polypeptide chain information. ठीक है, whereas the other secondary structures like beta sheets and the tertiary interactions, they can only form once the polypeptide exits the larger ribosomal subunit. So once it is present or once it is released into the cytoplasm, then only the polypeptide chain is going to form into a protein. Okay, so protein means it is having the secondary, tertiary, or last, if it is required, the quaternary structures. Okay. Now, but what it is saying is that in contrast to mRNA channel, okay, mRNA entry channel, the exit channel of the peptide chain, it is wider, more wider. Okay, अगर mRNA का इतना है तो polypeptide chain का इतना ज़्यादा है. Why? Because there are chances that the polypeptide chain is carrying the information, okay, information for forming alpha helices. So those polypeptide chain like N terminal है आपका, alpha helices ऐसे से form हो सकता है, and then you can have normal ऐसे structures. So these structures can form, and to allow proper exit of these structures, the channel of the polypeptide chain, okay, exit channel for the polypeptide chain is wider as compared to your mRNA channel. ठीक है, whereas the other secondary structures like beta sheets हो गया, tertiary structures हो गए, ये सब आपका बाद में हो सकता है once it is released into the cytoplasm. Guys, is it clear? This slide is clear? Yeah,
Yes,
so look it over here. So you have got the EPA, sorry. So here we have got the EPA, exit, peptidyl and amino, and then you can see the 50S over here. So very nicely you can see this gap which is present. तो जैसे ही P में आपका polypeptide chain आता है, from here it starts removing. ठीक है? ये भी कैसे दिखाया है? 50S नीचे दिखाया है, आपका tRNAs यहाँ दिखाया है. So just reverse the structure and you can visualize them. कि कैसे आपका P जो है यहाँ पे present है, A जो यहाँ present है, E ऐसे present है. Okay? So now you can just visualize how these polypeptide chains are finally exiting from the channel. Okay. So finally, now we are going to start with our translation steps in the upcoming lectures. So first, क्या होगा आपका? Small subunit is going to interact with the mRNA and the initiated tRNA. Then only it gets recruited. Then only your large subunit gets recruited. Okay, and that's called the initiation of the synthesis of new polypeptide chain. यानी कि translation initiation. Then what happens? You have got the new aminoacyl-tRNA tRNAs coming and bringing the all your correct charge amino acids, and then the polypeptide chain is being formed. That is elongation. अगर आप notice करो, हमने replication के time पे, transcription के time पे, we haven't talked about elongation factors, you know, their role in detail. But in the case of translation, we have got those elongation factors and playing very crucial role. ठीक है, because they are responsible for your removing the for movement of your mRNA. Okay. And then the termination of the polypeptide sequences. Okay. Yeah. So maybe in the evening we can start with the initiation of the translation. Okay. So आज के lecture में कोई doubt है, guys? Yes. Anyone? Tanvi, Shruti?
no, ma'am.
Okay. So I'll be stopping here now.